Mitosis. Mitosis is the exact copying of cells. This is necessary for the repair, replacement, and growth of cells in most organisms. The cell cycle. The cell cycle is separated into three parts: G1, S, and G2. Altogether, they are called interphase. During G1, the cell increases in size and continues with its normal functions. During the S phase, the DNA is doubled in a process called replication. In the G2 phase, the cell continues to grow and extra organelles are produced as the cell prepares for division. Once the cell goes through the cell cycle of interphase, it comes to mitosis. Prophase is the first step of mitosis. During this phase, the nuclear membrane and nuclei disappears. The chromosomes will begin to thicken and shorten, and the centrioles will start to move to opposite poles, and the spindule fibers will start to form. Metaphase is the next step. Here, the spindle fibers will begin to pull the replicated chromosome to the equator, and they will line up randomly along the equatorial plate. Anaphase is the shortest stage. During this stage, the sister chromatids will separate as centromeres separate. Each of the chromatids are now called chromosomes. The spindle fibers will then pull the chromosomes towards the poles. The final phase of mitosis is telophase. Here, a nuclear membrane will start to reform, and two nuclei will now form. The chromosomes will become less condensed, and cytokinases will occur via a cell plate. Cancer. Cancer is caused when your cells will no longer properly divide. The cells will begin to divide too fast and will continue to divide even if the requirements of cell division are not there. These cells move very quickly through the interphase, therefore they do not properly form and will be unable to do their jobs. The cancer cells start to use up the space and food of the healthy cells. So these cells cannot help the rest of the body properly function. These cells can also break away from their original places and move to other parts of the body. This is called metastasizing. Normal cells versus cancer cells. Normal cells have a much larger cytoplasm. They have single nuclei and fine chromatin. Cancer cells have a much smaller area of cytoplasm, and they also have multiple nuclei and coarse chromatin. The death and aging of cells. Cells have limits to the amount of times it can divide in a lifetime. The cells run on a biological clock, and once they are unable to divide, they will start aging and then die. Telomeres. Telomeres are repeating DNA sequences found on the end of the body's chromosomes. The purposes of telomeres are to prevent chromosomes from losing base pair sequences at their ends. Every time a cell divides, some of the telomeres are lost, and once the chromosome hits a critical length, they will no longer divide and they will die without their daughter cells. Telomerase enzyme. This enzyme elongates the chromosomes by adding TTA-GGG sequences to their ends. This enzyme is found in adult gametes, fetal tissue, and cancer tumor cells. Telomerase activity is regulated through development and has a very low activity in body cells. Since these body cells don't regularly use telomerase, they age. If telomerase is activated in a cell, that cell will continue to grow and divide.